We welcome Dr. Guilhem Tourniare, Managing Director at CELLENION, a spin-off company of CELLENION based in Lyon, specialized in single-cell and 3D cellular models isolation with its groundbreaking CELLENION technology. In 2020, CELLENION and CELLENION were acquired by the BICO Group, the world leader in bioconvergence. Recently, CELLENION released spheroONE, a unique platform for sorting and isolation of spheroids and organoids. This technology plays a crucial role toward implementation of 3D cell assays in drug discovery and their improved reproducibility. Guilhem has got a PhD in high-throughput chemical biology from the University of Edinburgh and has worked on the development of biosensors for glucose monitoring and of biomaterials for stem cell culture and bacterial enrichment before focusing on single-cell applications at CELLENION. In this webinar, Guilhem will present the method of high-throughput formation of hepatic spheroids using differentiated HepaRG cells developed in collaboration with BioPredict. How these spheroids can be isolated using spheroONE and what is the impact of this procedure on the morphology, viability, and functions of these spheroids. Guilhem, go ahead, please.
Thank you very much, Pamela, for the introduction and the invitation. Can you just confirm that you see the full screen mode, presentation mode? All good?
Yes, perfect.
All right, let's start. So my presentation will be much less biological than Leo's previous presentation. So it's more technology that we are focusing on at CELLENION. So first of all, thank you very much for inviting me to do this. Our mission at CELLENION is to provide disruptive technology, product, and services to master our cell biology. It's a big undertaking, I admit, but that's the mission statement that englobe the technologies that we have developed. So historically, we first developed the CELLENION technology. It's a technology for single-cell isolation that is very versatile. It also includes nanoliter dispensing, and this technology is widely used in the field of single-cell omics and cell line development. We can handle a wide range of particles with this, from the size of bacteria all the way to very large mammalian cells, such as cardiomyocytes. More recently, we've developed the spheroONE, which I will tell you a bit more later. But just a few words on the CELLENION technology. We've also adapted it to certain user requirements. So we have different versions. We have the standalone version, CELLENION X1. We have a BSC integrated version for handling of pathogenic sample. And for the pharmaceutical industry, we have also developed the HT version, which is to be interfaced with other robotic devices in order to undertake fully autonomous cell line development. We are very strong in the field of single-cell proteomic sample preparation. So we use the device to isolate single cell, but also dispense reagent, lysis, and digestion buffer. We have developed a range of different consumables, which are nanowells, which are used for improvement in the preparation of those samples prior to mass spectrometry analysis. So if you're interested in this, contact me after the webinar, but today I will focus on our new device, which is called spheroONE, which has been designed to sort and isolate spheroids, organoids, and tumoroids. So we can handle particles ranging from about 100 micron to 600 micron with technology. So first, regarding the technology, why did we develop this? Well, we believe from what we heard before and from discussing with many researchers that those 3D cell models are very important to replace animal models, as you know, but they tend to be very difficult to manipulate. They can be fragile. The way that they are produced sometimes makes their imaging very tricky and readout of assays. Many of the workflow have a fairly low throughput using, for example, ULA plates. And also what is seen is using those ULA plates, you don't always get very homogeneous spheroids formed at the bottom of those. They can be heterogeneous in terms of size, in terms of the biomass present, and also their functionality. And this sometimes leads to poor assay reproducibility and making the interpretation of the results difficult. So our solution has been to develop this spheroONE device in order to allow the sorting and isolation of those spheroids in order to obtain very homogeneous population for your assays. The technology, spheroONE technology, is based on a nanoliter dispenser. So we have these glass capillaries that are connected to an electromagnetic valve. And with those, we can produce very reproducible droplets, ranging from about 100 to 500 nanoliter. And this capillary is mounted on an XYZ axis with which has a very high precision, which allows us to dispense those spheroids or organoids into a range of different substrates. So this can be your traditional 96, 384 well plate, even the 1536 well plate, or we can also isolate those on slides to facilitate imaging of those or in custom substrate. But so we generate droplets. Obviously, if you want to sort and isolate one at a time, you need more than that. So what we have developed is like some algorithm and imaging capabilities. So what we do is we look down the tip of the dispenser, and basically we segment this dispenser, the tip of the dispenser, in two zones. The ejection zone, which corresponds to the volume of one droplet, and we have also a buffer zone above, which we call the sedimentation zone, to account for the fact that these large particles tend to sediment fairly quickly when nothing is happening. Then how does this image-based technology work? Basically, we image the tip of the dispenser, and whenever we will detect the presence of one particle in the ejection zone, we know that in the next droplet that we will generate, we will eject the droplet containing that particle. So if that happens, we move the dispenser from the camera position to your plate, the first well of your plate, dispense the droplet containing the spheroids. Go back to the camera, take again an image. In some cases, in many cases, the ejection zone will be empty. In that case, we generate another droplet until we have a particle coming in the ejection zone. We will have cases where we have two particles. In that case, we discard them. We don't want two, we want a single one. Or also sometimes we can have a particle in the sedimentation zone and one in the ejection zone. In that case, we also dispose them. When I say we discard them, a particularity of our technology is that we don't put that in a bin. We put that actually in a small tube. So you have the ability to take again those discarded spheroid and organoid and process them again. It's an image-based technology. So we take a number of measurements on the particles that are seen in the ejection zone. We measure their diameter and their circularity. And obviously, being image-based, we have an image of every particle that is isolated, and we know in which well it is isolated, which can be very useful for your quality control. The modality we use for imaging those particles is called dark field imaging. So it consists of shining a light at an angle, and basically what is collected by the objective is the light scattered by the particle. And the image we get are what you see on the right. So we see a white image on a black background for the particles that are going through the capillary. How do we use this spheroONE technology? Well, as I said, we use it for isolation of single spheroids. So in this example, we started from a very heterogeneous bulk spheroid preparation, which we prepared by liquid overlay. So just shaking, agitation, incubation under agitation over several days. You generate a wide range of sizes of spheroids, but also typically in those methods, you will have a lot of debris. You can generate thousands and thousands of spheroids, but yeah, they will be heterogeneous in size and they will present a number of dead cells and debris. But now we load this suspension into the spheroONE, and then we're going to isolate one by one spheroids into a 96 well plate, for example. And here is an example where we did like 4 plates with 1 spheroid per well. We obtained an accuracy about 91%, and it took us about 4 minutes for each 96 well plate to prepare. Another thing we can do is we can sort by size. So we can start from, again, an heterogeneous population, gate from 100 to 500 micron. This is what we obtain in blue. We can also have a gate for just the largest spheroids or the smallest spheroids. In that case, with a very narrow gate of 200 to 250. And you can see that all of the isolated spheroids looks very similar. We can dispense one, but we can also dispense multiple spheroids per well. So in that case, we did 1 spheroid per well, 3 spheroids per well, 5 spheroids per well. And this allows you to do like a control of biomass. We can also dispense those spheroids, as I said, with very high precision, so we can position them in micro devices. This is a chip that we have developed, which has a diameter at the bottom of 400 microns, so we can easily position the spheroid at the bottom of this well, or we can imagine bringing different spheroids to do co-culture of different spheroids. And also an application that some of our customers are doing is arraying of organoids on surfaces in order to facilitate their imaging so that they're all in the same plane. We have had a collaboration also with UPM, which is a Finnish company that developed the Grodex. Grodex is an hydrogel made of nanocellulose, sorry. And in this collaboration, we wanted to see whether our spheroONE was going to be compatible with the dispensing of the hydrogel and also the sorting and isolation of spheroids suspended in the hydrogel. So in this study, what we did is we first coated the well of the plate, of the 384 well plate, with a layer of Grodex. Then we isolated single spheroids of HepaRG spheroids, added another layer to completely embed the spheroids, and then added the culture media on top. And this worked very well for concentration up to 0.5%. In the field of organoids, it's very important, as you know, many protocol uses matrigel for the differentiation and maturation of the organoids. So here we have to adopt a slightly different methods because the matrigel is very viscous, so we cannot dispense it directly with the spheroONE. But in the spheroONE, we can control the temperature of the plate. We have a temperature control holder for the microplate in which you're going to deposit your particles. And so what we do is we pre-fill the well with matrigel at 4 degrees. Then we keep the plate at 4 degrees while we do the isolation of the spheroids or pre-organoids and organoids. At this temperature, the matrigel is still liquid. So when the organoid reaches the gel, it goes into the gel. Then we bring the plate, after isolating all the organoids, we bring the plate at 37 degrees, which induces the gelation. And then we can just add culture media on top. And here you can see some example of organoid maturing over 5 days. We have used this approach in collaboration with some academic partner here in Lyon at the Center Léon Bérard with Cédric Chavroux, where we isolated lung spheroids into matrigel, and we did the same with kidney organoids with the Gemelli Hospital in Roma. So this gives you an overview of this technology, of the spheroONE technology. In summary, what does that do? It allows you to have accurate isolation of single spheroids. It allows you to have highly homogeneous plates by selecting specific subpopulation or user-defined sizes. You can also control the biomass if that's important for your assays. Given that we don't get all the debris and the dead cells that are sometimes present at the bottom of ULA plates, it facilitates also the imaging and assay readouts. And it can also be used for organoid selection by selecting specific sizes of pre-organoids and letting them mature in matrigel. Now I'm going to focus on more of the work that we did in collaboration with BioPredict. In this collaboration, we used the HPR116 HepaRG cells, which are the differentiated HepaRG cells. So traditional methods, as we've seen before, tend to be in ULA plates. In these methods, they are quite tedious. You have to pipette in each well a certain number of cells, wait for several days for the formation of the spheroids, which can take quite a lot of space in your incubator for several days. And typically, you can obtain highly heterogeneous spheroid size, and also, as I was saying before, debris and dead cells at the bottom of those wells. So the idea of this study was to develop a method, first to have a method for producing high number of spheroids, then demonstrating that with the spheroONE we could sort them and isolate them. And finally, we did a few just initial functional testing on those isolated HepaRG spheroid to demonstrate that they were good for further application assays. So the method, we tried a number of different methods, but the method that works the best in our hands was using microcavity plates. So these are manufactured by Corning. There are a few different manufacturers of microcavity plate like Kugelmeier and Corning. Basically, what you have is in a 24-well plate format, at the bottom of the well, you have 500 microcavities. So you will generate 500 spheroids in each of the 24 well. So you can generate a large number of spheroids just from one plate. After 4 days of formation at 37 degrees, we collected those spheroids. They were nicely packed. We measured their diameter, so they were ranging roughly from about 150 micron to 350 micron. We loaded this into the spheroONE reservoir and then isolated only the spheroids with a diameter ranging from 200 to 250 micron. We obtained something, well, first it worked. We obtained plates, a 384 well plate filled with one spheroid per well. We could see that by measuring their diameter under the microscope that the set diameter on the spheroONE were respected, and we obtained only spheroids with diameter, with a desired diameter. We did a few live dead experiments on some of those spheroids, verified that no dead cells were present. As you can see here, we had a very good live dead staining with no dead cells present. And then we undertook some albumin secretion assays on the supernatant for a few of those spheroids, and also did some ATP production assays. And all these confirmed the functionality of those HepaRG spheroids. So in summary, for this little study, we developed a high throughput formation method for HepaRG spheroids. We demonstrated that spheroONE was suitable for sorting and isolation of homogeneous spheroid population. We verified the integrity of the spheroid post-isolation and their viability, and also their functionality. Overall, we have an efficient method for the preparation of HepaRG spheroid assay-ready plates. And in the future, we still have to do some functional validation, and we are contemplating potential commercialization of those assay-ready plates filled with a single HepaRG spheroid. So if you're interested in this technology or acquiring some plates, don't hesitate to contact me afterward. Finally, we'd like to thank, well, you for attending, and our collaborator at Biopredict, so Agnès Jamin, who has been heavily involved in this project, together with Solène Pasquier, who is our research scientist at CELLENION, Pamela for organizing this nice webinar and inviting us, and Christophe for all the exchanges over the year, and at CELLENION, Stéphane Cosson, who has been the project lead on the development of the spheroONE technology, and Cécile Thion, who helped me putting together those slides with our product manager for the spheroONE. If you'd like to contact me, this is my personal email address, g.tourniare@cellenion.com, and you can also contact us at contact@cellenion.com, or follow us on our different stream. Thank you very much.
Thank you, Guilhem. Thank you for this nice talk. Now, Guilhem is ready to take your questions. The first one, have you ever embedded this spheroid for subsequent histology, cutting paraffin slice, for example, with precise Z-positioning?
So the answer is no, we have not. These are not methods that we have currently internally at CELLENION, but we are happy if you want to send us some spheroids that we then embed for this histology and cutting. That's absolutely something we could do, yeah. But we have not done it yet at CELLENION.
Okay. Guilhem, what is the range of size of spheroids that spheroONE can sort and isolate?
So we can handle any particles from 100 micron to 600 micron in diameter.
Okay. Thank you. And what about, so you said that you screened the spheroids for albumin secretion and the ATP. Did you look over the expression of some cytochromes or transporters, if it is enhanced in these spheroids compared to 2D culture?
No, we have not done that. So in the past, we did a study, but these were with the undifferentiated HepaRG, where we compared, so we did a single-cell transcriptomic study with the CELLENION device in the past, where we compared 2D culture of undifferentiated HepaRG versus a 3D model, a spheroid made of those. And we could clearly see that the spheroids had a much more hepatocyte-like phenotype than the 2D culture. But we have not done those experiments.
Okay. And is it possible to collect spheroids from the microcavities of the plates?
Yes. So the way we collect them is just by pipetting them. So you just agitate a bit in the microplate, and you can easily collect all the spheroids that are present in the well and in the microcavity.
Okay.
Fairly straightforward.
Mm-hmm. And do you think that spheroONE is suitable for co-cultured spheroids like the ones presented by Leo?
So as long as the spheroids is generated, then if it fits within the size range that we can handle, yes, absolutely, we can isolate those.
Okay. Very practical. Yes, sorry.
Yeah, we don't. So the spheroONE is not used for the preparation of the spheroids, but as long as you provide us with spheroids that are, yeah.
Yes, it should work. Okay. Very practical question. What is the time required to isolate 96 spheroids using spheroONE?
So for a 96 well plate, it typically takes us about 4 to 5 minutes for 96 single spheroids.
Okay, good. Has spheroONE been used in other applications than drug discovery?
So we have a collaboration with an academic partner here in Lyon who are using it for positioning of chondrocyte organoids. And the idea there, so they put it in kind of a mesh scaffold, and they want to reconstruct. So it's for tissue engineering application where they want to reconstruct cartilage tissues.
Okay. Okay, and what about the longevity of your spheroid? Did you check this parameter?
Yeah, this we have not checked how long we could cultivate those. I think this will be part of the future experiment where we have to see the longevity of those. We have not tested their viability after like 5 days post-isolation, but there is no reason to believe that they would not survive for much longer.
Yes, maybe it depends on the cell type because the morphology, the ATP and the albumin secretion remain the same. So maybe it more depends on the cell type rather than the spheroONE technology.
Mm-hmm.
Thank you, Guilhem.
Very welcome. Thank you.